We'll be looking at the topic of the control of gene expression in the eukaryotes. Okay, earlier you have learned about the control of gene expression in prokaryotes with the lag and trip operon. So now it is for the eukaryotes. Alright, so we'll be first looking at the control at the chromatin level, which controls the level of packing of the chromatin, whether the chromatin is tightly packed or loosely packed, and that will in turn control whether the genes found in that region of the chromatin, whether they are transcribed or expressed or not. Alright, so the level of packing of the chromatin, right, affects transcription and gene expression because it determines whether the proteins involved in transcription, whether these proteins can access the DNA or not. So for example, right, you have learned about the RNA polymerase as well as the general transcription factors that must bind to the promoter, right, to form the transcription initiation complex. Okay, so if the DNA is closely packed or the chromatin is closely packed, then these proteins, right, would not be able to bind to the promoter because of the tight packing, right, and therefore there's no transcription. Okay, so to control transcription, right, the first level would be to control how tightly packed the DNA is at that region of the chromatin where the genes are found. Okay, so recall that there are two kinds of packing in general, right? For chromatin, you have the euchromatin, which is less condensed or less tightly packed, versus the heterochromatin, which is more tightly packed. Right, so you should be able to recognize the euchromatin and the heterochromatin in the picture of the nucleus. Okay, so the darker region would be the heterochromatin and the lighter region would be the euchromatin, as you can see here in this diagram. But when I say darker region, which refers to the heterochromatin, right, please do not confuse the darker region with the nucleolus. Right? So the nucleolus is a bit different. Yeah, nucleolus is not consisting of heterochromatin. Okay, even though it is like very dark or very darkly stained. Okay. Recall that the nucleolus contains genes coding for rRNA, so obviously every cells make ribosomes, so the genes for rRNA would be transcribed in most cells. Okay, so that wouldn't be the heterochromatin, yeah. But other than the nucleolus, right, the other darker region that you can see in the nucleus, those would consist of the heterochromatin, which is transcriptionally inactive, right? The genes found in the heterochromatin would not be expressed. All right. Okay, so let's continue to look at how can the cell controls the extent of packing of chromatin. Right. So the first method is the acetylation of histones. Okay, so earlier you learned about how the DNA is wound around histones to form nucleosomes. And the winding of the DNA, right, to the histones is because the DNA is negatively charged and histones are positively charged. Okay, so when there is the acetylation, which means the addition of acetyl groups, right, to the histones, and specifically to the lysine residues at the N-terminus of histones. The lysine residue, if you look at the R group under the protein notes, the lysine residues, the R group is positively charged. Okay, so when we add acetyl groups, right, to these lysines, it blocks the positive charge of the R groups of lysines. So overall, right, the histones proteins become less positively charged. Right, it is still like positive, right? There will be still other, positively charged R groups around. If not, the DNA wouldn't be able to wind around histones, right? But then the overall charge is less positive. Okay, so the binding between histones and DNA would be less tight, and therefore the chromatin is less tightly packed or coiled. right, the DNA wouldn't coil around the histones so tightly. So overall, the chromatin structure kind of loosened up a bit, and that forms the euchromatin structure. So the reverse, when we remove the acetyl groups from histones, that's the histones deacetylation, right? Then the positive charge of the histone proteins would be restored, and therefore the binding between DNA and histones would go back to the usual tightness, and therefore the tighter coiling of the chromatin to form the heterochromatin structures. Okay, so the enzymes, right? Obviously, this acetylation and deacetylation are catalyzed by enzymes. Right, histone acetylation is catalyzed by histone acetylase, and histone deacetylation is catalyzed by histone deacetylases. Right, so pretty obvious names. Right. Okay, so that's one way how the cell can control or change the packing of the chromatin. Another method is to modify the DNA instead of the histones. Right, so DNA can be added with methyl groups, and usually this happens on the cytosine nucleotides, or rather the cytosine bases, right? And not any cytosine bases, right? Usually it will be at the CPG sites or CPG islands. Okay, the CPG here refers to, if you look at my cursor here, right, the CPG would refer to the sequence of a C base and a G base. So in between the nucleotide is a phosphate, right? That's why it's a CPG, right? So usually C and G together, the C can be methylated, okay? And that is catalyzed by DNA methyltransferase, which, as you can see here, transfer the methyl group CH3 right, to the cytosine base. Okay, so this DNA methylation, right, has two effects. Number one is that it can directly block the binding of certain transcription factors, because these CPG sites, right, are very common in binding sites of certain transcription factors, not the general transcription factors, because the general transcription factors binds to the promoter, right, which is and they recognize the TATA box, right, TATA, right. So that's not recognizing the C and G, but there are other transcription factors which you will learn a bit later, which binds to other sites on the DNA, and some of these sites, right, on the DNA would contain a lot of these CPG islands or CPG sites. Okay, so the binding or the addition of methyl groups, right, at these sites, right, can directly block the binding of certain transcription factors, which will affect the initiation of transcription. Okay, and secondly, right, the methylated DNA is also recognized by the enzyme histone deacetylase, which we have looked at just now, right? So that will induce the formation of heterochromatin at that region of the DNA, and therefore whatever genes you find in that region, right, would be transcriptionally inactive. Okay, so two effects of DNA methylation. One directly affecting the binding of transcription factors, the other kind of indirect through the formation of heterochromatin and reducing the excess of the DNA, right, by the proteins. All right. So yeah, just kind of summing up two different methods. One to modify the histones through acetylation, the other to modify the DNA through methylation. Right, so the euchromatin, which is loosely packed, then the histones would be acetylated and the DNA wouldn't be methylated. right? As for the heterochromatin, the DNA is methylated, which in turn induce histone deacetylation. Okay, so a bit of a summary, right, in a table. Okay, so you might want to take some time to try to summarize what you have looked at yourself first before you continue with the video to look at my answer. Alright, so you may want to pause the video now. Okay, so if you have done your own summary and consolidation in this table, you can check with my answer. Right, so for histone acetylation, it happens at the histones, obviously, and not anywhere in the histones, but specifically the lysine residues as the N-terminus. For the DNA methylation, of course, occurs to the DNA, but specifically at the cytosine at CpG islands or CpG rich sites. All right, chemical group added, obviously acetyl groups versus the methyl groups. Effect would be opposite of each other, right? The histone acetylation forms euchromatin and DNA methylation forms heterochromatin, and therefore the effect on gene expression is also kind of opposite of each other, right? Histone acetylation allows for a higher accessibility of the DNA to the proteins to initiate transcription. But for DNA methylation, it lowers the accessibility and also directly block the binding of some transcription factors. So therefore, there's no initiation of transcription. All right, so that would be the first part on the gene regulation at the chromatin level.